Okay. Well, good afternoon, everyone. There's a lot of people online, and welcome to Pediatric Teaching for today. Last week, we talked about genetics in pediatrics, and we had an overview of the history of genetics and some other aspects of genetics. I'm going to talk a bit more about the some practical aspects of genetics today, some basics that you need to know, especially for the Part 1 examinations. And so it's good to look at this lecture in the context of the one I gave last week and then put them together. And I think you'll know all you need to know about genetics, or I hope so anyway. Okay, this is what we're going to talk about today. Firstly, the basics. So I'm going to talk about the organization and processing of genetic information. And what that means is how genes are organized onto chromosomes, where they are in the cell, in the nucleus, and also in the mitochondria, and how they are transcribed and translated into messenger RNA and then into proteins, amino acids combined into proteins, and how all that works. And so, because this is something you really need to know for the Part 1 examination in all disciplines, actually, but certainly in pediatrics. We talked a bit last week about the ways to identify genetic disorders, because I showed the various ways in which we can view chromosomes through G-banding and through fluorescent in situ hybridization. And I talked a bit about some more modern methods that have helped us understand genetic disorders. I'll just briefly mention them again today. I want to talk a bit about mutations and when they occurred. This is quite important for understanding different types of genetic disorders and how genetic changes that occur after embryo formation and during life impact on other diseases that we may get. So I'll talk about mutations, both those that are inherited, but also those that occur afterwards. Last week we talked about Gregor Mendel, the Austrian monk who grew peas and identified the first inheritance patterns of autosomal dominant and autosomal recessive conditions. And I'll talk a bit more in detail about these single gene defect Mendelian inheritance patterns, because these are the sorts of things you need to know about: autosomal dominant, autosomal recessive, X-linked conditions, etc. I'll briefly mention mitochondrial inheritance, because there are genes, of course, both in the nucleus but also in the mitochondria, and they can code genetic mutations in those genes, can code for certain diseases. And I'll just briefly mention that. We'll talk a bit about the regulation of genes, because not all genes are active at all the time, and they're regulated by by a variety of different changes in genetic information that can lead to a gene being active or being suppressed. And the sorts of things that make genes more active or more suppressed and not active are partly normal processes, and they're partly abnormal processes. And we'll talk a bit about the abnormal processes particularly, and that's a field called epigenetics. It's how our environment might change our genetic expression and lead to different conditions. And then lastly, almost lastly, we'll talk about just some common congenital disorders in children in Papua New Guinea. And I just want to point out that most of those are not directly inherited. They have a genetic basis, but it's what it's called polygenetic. That is, there's a variety of different genetic mutations that can contribute to the development of these conditions. I'll go through them in a bit more detail. And then I just want to finish off by trying to address the question of why I learned about genetics, but I hope by the end of it you'll have a good understanding why you should learn a bit about genetics. Right. So just to start with the real basics of genes, I think you know this, but genes are all encoded in DNA, deoxyribonucleic acid, that's in the nucleus of the cell. There are 4 what we call nucleotide bases in DNA: adenine, thymine, cytosine, and guanine. And a sugar molecule joins them up, and they form this this helix, a double helix, where you've got adenine and thymine joining, and cytosine and guanine joining, and they're joined by this sugar molecule, deoxyribose, that links the molecules, and they form this helix, this double helix. The hydrogen bonds link the double helix, and there's this sugar phosphate backbone that makes the helix. That's really all you need to know about that. There are two locations, as I mentioned, where genetic material exists within a cell, and the main one is in the nucleus, where there's 3 billion nucleotides, and that codes for more than 25,000 genes. So almost all our genetic information is within the nucleus of the cell, but there are some genes that are in the mitochondria, and I'll talk a bit more about those. Just 37 genes encoded in mitochondrial DNA, but they're quite important genes in terms of regulating the respiratory chain enzymes and energy production, and genetic mutations that occur in mitochondrial DNA can lead to some of the chronic diseases of adulthood that affect major organ dysfunction, such as chronic cardiac disease, for example, could sometimes relate to mutations in the mitochondrial DNA as we get older. The DNA is tightly packaged into 23 chromosomes. We went through the karyotype last week. That is, there's 2 copies of each chromosome, 23 pairs, and that's 46 chromosomes, so 22 autosomes and 2 paired sex chromosomes. Remember, the sex chromosomes are XX or XY. I'm sure you know all that, and they're encoded within. They're packaged within DNA that occurs within the nucleus of the cell. As I said, the mitochondria plays an important role in providing energy for all organs, and it provides a source of adenine triphosphate, ATP, for aerobic metabolism. And ATP is the source of most of our cellular energy function. But remember, I said there was only 37 genes that code for 13 proteins in mitochondrial DNA, and but these tissues, these mitochondrial tissues, are highly metabolically active, and in certain organs within the body we have more mitochondria. So in highly metabolically active cells, like in brain cells, we have more mitochondria. All the interesting genetic dimension to mitochondrial DNA is that it's all maternally inherited. So you don't get mitochondrial DNA from your father and your mother like you do the DNA that occurs in your nucleus. You get all the mitochondrial DNA from your mother, maternally inherited. And so that's why some conditions that are maternally inherited can be mitochondrial conditions. I'll talk a bit more about that a little while. The main thing about DNA and genes and proteins, you just need to know how DNA encoded in chromosomes is transcribed into RNA and then converted into messenger RNA and then translated into amino acids, an amino acid chain that forms a protein. That's basically it. A gene's a series of nucleotides. Remember we said adenine, thymine, guanine, cytosine. Some genes will have like 100 or 1,000 nucleotide bases. And as I mentioned, in the nucleus of a cell, there's about 3 billion nucleotides, and that encodes for those 25,000 genes that make us what we are. Remember I said DNA is transcribed into RNA, and then that's further transformed into messenger RNA. I'll explain how that happens. And then it's translated into an amino acid chain that forms a protein. Within the DNA, there are what we call coding sections of the DNA. They're the exons of the DNA. So in this schematic diagram, the exons are blue, and the introns, which are intermediate regions within the DNA, are red. And DNA is transcribed to RNA, and that has both exons that is the coding regions of the genes, and introns, which are I would think consider them the intermediate regions. And that the introns have regulatory roles in gene expression. So they can turn genes on or turn genes off, but they're not, once RNA is transformed into messenger RNA for transportation out of the nucleus onto the endoplasmic reticulum, where amino acids form the proteins, then the introns are spliced off. So that's the step between RNA and messenger RNA. So messenger RNA doesn't contain introns, whereas RNA contains both exons and introns. Remember, the introns are the regulatory regions, or they have some sort of ancient evolutionary role, but they're not coded into amino acids and into proteins. Mitochondrial DNA that I talked about before, that very minor form of DNA that we have within our mitochondria, they don't have introns. And as I said, neither does messenger RNA. Messenger RNA only has exons, and that's what's translated into an amino acid chain and forms a protein. RNA is single-stranded. It's not a double helix like DNA. And in RNA, instead of thymine, adenine, thymine, guanine, cytosine in DNA, then the thymine is substituted for uracil. So it's adenine, uracil, guanine, cytosine in RNA. The main thing to know is that when RNA is spliced, the introns are spliced out, that becomes messenger RNA, which then forms the template for the protein polypeptide within the cell. We have talked a bit about this, that the amino acids link together. That's in the endoplasmic reticulum. They form a three-dimensional shape, and there are certain regions of the DNA. One's called stop codons that indicate the end of a gene or the end of the translation of amino acids from messenger RNA to an amino acid chain. Most of the genome, the human genome, are what we call highly conserved. That is, they're almost always translated into proteins, but sorry, not most of it. A small proportion, only 5%, are actually translated into proteins, and 95% is much less conserved. And that means we don't really know what the function of those non-coding regions are. Those non-coding regions of messenger RNA, not the introns, but there are many non-coding regions that actually aren't translated into amino acids. But the function of those is not entirely certain. They certainly have some function, but it's not entirely certain. And these days, when you do what we call a whole exome sequence, where you can look at every gene, then there's a whole lot of changes that can occur in DNA that's actually not translated into amino acids, and we have no idea what that actually means. It probably doesn't mean anything serious in terms of disease for most of the cases. As I said, there are ways of identifying genetic disorders, both old ways and new ways. And the oldest way, remember I said last week, is using what we call cytogenetics, or looking at a carrier type within a cell. These were first identified in tumor cells, if you remember, way back in the early 1900s. And a carrier type where there's G banding, that black and white, that grey and white banding of the chromosomes. That was the earliest way in which major structural changes in chromosomes could be identified. And now, more recently, well, in the 1970s, actually, there was this technique called fluorescent in situ hybridization, which enabled sort of color banding instead of the black and white or banding of chromosomes. And it was possible to see smaller and smaller changes within chromosomes. In the last 20 years, there's been these techniques called microarrays that enable the detection of smaller and smaller deletions and duplications that you can't see with traditional cytogenetic analysis of cells and the karyotype. A microarray can detect single gene disorders, very small copy number variations, and mosaicism. Remember, mosaicism is where you've got some cells that actually have a mutated form of the genetic code and some that have a normal form of the genetic code. That's a mosaic. And microarrays can detect that. And now we have this type of technique in some countries called whole exome sequencing, and that detects every genetic disorder that can be identified. It detects single gene disorders down to a very, very microscopic level, and changes in DNA sequences. But remember, because there's so much of the genetic code that we don't quite know what it is, then you can find gene deletions or mutations in some of the genetic code, and you don't, at the end of the day, you don't quite know what the significance of all of that. I want to talk a bit about mutations, and because they're the basis for all genetic variation, good ones and bad ones. Not all mutations are bad. You know, the evolutionary path that has led to a whole range of changes have occurred mostly because of random mutations that are beneficial for survival. Mutations occur at various phases, but often in mitosis. That is the process of copying DNA and dividing it into two equivalent cells. That's when cells divide, there has to be a process of mitosis that provides an exact copy of the DNA from one cell to make then two cells, and two cells to make four cells, and four cells to make 8 cells, etc. There is an enzyme called DNA polymerase that does this copying of the DNA in the process of mitosis, and the error rate of DNA polymerase in mitosis is about one nucleotide pair per million copied, and it might seem very low, but actually by the time we reach adulthood, we've usually got hundreds of new errors that have occurred because of errors in mitosis, and each error then becomes evident in all the descended cells. So if you've got an error in the cell that's been copied, then every cell that's copied after that will have that error, and that's the basis for what we call genetic polymorphism, or changes in DNA sequence that some of which, not all, but some of which may have a pathogenic change, and some of which might have a potentially even an evolutionarily beneficial change. But mostly we go looking for pathogenic mutations rather than changes in the DNA structure that have some benefit. Now, I want to just describe when mutations can occur. This is important because they can occur like during the formation of ovum or sperm. Now, if they occur during the formation of ovum and sperm, then that will be transmitted to the fetus, of course, and more than half of all human pregnancies fail by 6 weeks, and most of that is caused by new mutations in gametes. Usually that failure of conception usually occurs before a woman knows that she is pregnant. But many of these new mutations are caused by new mutations that occur in the ovum or the sperm in gamete formation. Now, if a mutation occurs during gamete formation in one generation, but the gamete is fertilized with an ovum or a sperm, and the fetus survives, that is then that condition or that genetic mutation will be transmitted to the next generation. Therefore, it becomes familial. So that's the very earliest type of mutation that can occur. The next mutation that can occur is during embryogenesis. So not at the fusion of the gametes phase, but sometime during embryogenesis. And if a genetic mutation occurs during embryogenesis, then it may be that only certain tissues are affected. So, for example, if it's a somatic mutation, it might only be affecting certain tissues, perhaps of the gastrointestinal tract or of the cardiovascular system, etc. And usually these types of mutations are not hereditable. That is, they're not transferred from a child or a parent to another child, even if the child survives into adulthood and reproduces, because it's not affecting it's not a gonadal mutation. It's not affecting the gonads. If there's a mutation affecting the gonads, then in that occurs during embryogenesis, that is potentially hereditable. That is potentially someone so affected may pass it on to a subsequent child. So we've learned about mutations that occur in gametes before they're fertilized, or even at the time of fertilization, and we've learned about mutations that occur during embryogenesis, most of which involve one organ system rather than the entire body. It depends how early in the phase of embryogenesis that it occurs. And then the third sort of mutation is after embryogenesis, like what happens after an embryo is fully formed. And I mentioned before that 1 in every million mitoses of the cells in our body, there'll be some sort of mutation. And throughout our life, then new mutations can occur, and they can be transmitted to the progeny of that cell, that is the subsequent cells. That they won't be passed on usually to subsequent offspring, but they may result in certain things like cell death, or sometimes they may result in disease. We talked last week about how almost all cancers arise from genetic mutations somewhere in after embryogenesis, and they may be triggered by environmental exposure. Smoking, for example, leading to lung cancer is all related to mutations. Skin cancer, melanoma, that's related to sun exposure, that leads to mutations, and then that can genetic mutations that can lead to the cancer. But again, they're after embryogenesis. I mentioned before that mitochondrial DNA is especially susceptible to mutations, and that doesn't, the manifestation of that, remember I said that mitochondrial DNA is really heavily involved in energy production within cells. It produces ATP for the Krebs cycle and for aerobic metabolism. If there's a genetic mutation that occurs after embryogenesis in some mitochondrial DNA, then that can lead to organ dysfunction and energy problems within cells, such as heart failure or dementia, something like that. Now, that's not the sole cause of adult chronic diseases, but it is partly a cause, genetic mutations that occur with age in mitochondrial DNA. Right. Now, I wanted to, so we've talked about mutations and when they can occur. I wanted to just go back a bit to talking about Mendelian inheritance, because this is often what we focus quite a bit on, Mendelian inheritance, where you've got either an autosomal recessive condition or a dominant condition. And I want you to be really clear about those types of conditions. Remember, they only explain single gene defects. Mendelian inheritance explains single gene defects, and actually most conditions are polygenetic, or they're environmental plus polygenetic. Polygenetic meaning that more than one gene is involved. But we're talking now about just single gene defects, and I wanted to just cover these types of Mendelian inheritance, which, remember, we said arose from those experiments that Gregor Mendel, the Austrian monk, made in the 1850s. Autosomal recessive conditions, there's a few characteristics of each of those that I just want you to know about. Autosomal recessive conditions affect both males and females. Usually the parents are not affected, because usually the parents are carriers, the heterozygous carriers. The affected individual has two copies of the recessive gene. They'll be little r, two recessive genes. That's because you need two recessive genes to have the phenotypic appearance. Whereas if you've got one recessive mutated gene and one normal gene, then usually the dominant gene will be dominant in the phenotype that's produced. We said that an individual such as both parents of a child with a recessive disorder will be a carrier. They'll have just one copy each of that genetic mutation. And the frequency of the genetic disorder, whether it's thalassemia or whether it's sickle cell disease, will depend on the frequency of the carrier status in that population. And so, as you know, the frequency of the beta thalassemia carrier status in some populations in the Pacific is actually quite high. It's related a bit to malaria endemicity, and the frequency of that is quite high. So therefore, there's a much higher prevalence of that disorder because there's a lot of carriers within the population. Remember, he said most of the male and female carriers, a parent, a father and a mother, will be big R normal and little r recessive disease, and both the mother and the father, and they'll have a 1 in 4 chance of having an affected offspring. So 2 in 4 will be normal, big R, and 1 in 4 will be recessive disease, and 1 in 4 will be a heterozygote. So that's 2 in 4 will be a heterozygote, 1 in 4 will be large R, and they won't have a recessive, they won't have the mutated gene, and 1 in 4 will actually have an affected offspring. The reason why recessive conditions are more common in some communities is consanguinity, and that allows a recessive condition to circulate around the population, and occasionally you'll have an affected offspring. Now, beta thalassemia is a common condition that is autosomal recessive, so you need to know quite a bit about that. Autosomal dominant conditions are, excuse me. Hello, it's Trevor Duke speaking. Yes, Sasha. Yeah, good, thanks. Yeah. Sorry about that. We'll keep going. So autosomal dominant conditions both affect, also like recessive conditions, affect both males and females, and they can be transmitted by males and females, but that usually the affected, the parent is also affected because it's a dominant condition. So it'll be a parent who's affected, transmitting the genetic disorder onto a child. And it can equally be present in both the mother or the father. If a parent is affected, they have a 50% chance of transmitting a dominant condition onto their child. And unlike with recessive conditions where both parents are not affected, but they have a 25% chance of transmitting their recessive condition onto their child. It's common in autosomal dominant conditions to find a family history of autosomal dominant conditions, whereas in autosomal recessive conditions, there might not be a family history. The affected individual, whether it's the child or the parent, is usually heterozygous. So if you're heterozygous with a dominant condition, then the disease is dominant. That's big R disease, and that overrides the normal or recessive condition, the normal gene, the normal gene in the cell. Sometimes you won't get a family history, that is, the parent won't be affected or will only be maybe mildly affected, and that's a phenomenon we call variable expression. and that can be variable penetrance, which means that it appears to skip a generation, or maybe there's a less affected parent having a more affected child, and that's just variable expression of the gene, the mutated gene in the offspring. Usually, I mean, always there'll be one parent affected, unless there's been a mutation that occurs during in what, remember we said, either gamete formation or embryo or gonadal mutation that occurs early in embryogenesis, and that can sometimes explain why a child with an autosomal dominant condition won't have a parent affected. So it could be what we call a new mutation, or it could be an inherited condition from an affected parent. That they're the two ways in which it can happen. There are a number of important autosomal dominant conditions. Von Willebrand's disease is quite common in the Pacific, and that's an autosomal dominant condition of coagulopathy and bleeding. There are some types of retinoblastoma that are also autosomal dominant, and they're the familial type, usually bilateral retinoblastoma. Not always is that inherited. That is, not always is there a parent affected. Sometimes it's a new mutation that occurs during gonadal formation, and therefore only the baby is affected, but the parents aren't affected. Achondroplasia, so that musculoskeletal condition or skeletal condition leading to very short stature is also an autosomal dominant condition. And if a person who has achondroplasia has offspring, then there's a 50% chance usually that the condition will be passed on. But mostly babies who have achondroplasia, their parents don't have achondroplasia, and that's because it's a new mutation that occurred during embryogenesis or right at the time of gamete fertilization. X-linked recessive conditions are important. They mostly affect males. X-linked recessive. Most of the X-linked conditions we know about are X-linked recessive. They mostly affect males. Very occasionally, females can be affected or have very mild disease, and they'll have very mild disease because they've got one normal X chromosome, one normal X chromosome in their cells, so they won't be strongly affected. But usually, males are much more affected. The X-linked recessive conditions are all transmitted from the mother, because from your father you get the Y chromosome, so that's not affected by the condition, but the X chromosome comes from the mother. So the family tree looks like the male child is affected, the mother is a carrier, but her father or uncles might also have been affected by the disease. So conditions like hemophilia, often you see a family history like that, whereas it appears to what we call skip a generation, but it just means that the genetic mutation was transmitted from a grandfather to their grandfather's daughter as a carrier, and then to her male offspring. That's usually the way. If you've got a family history of an X-linked condition, and that's usually the way it's transmitted. So the common X-linked conditions are Duchenne's muscular dystrophy. It's a very common, it's the most common muscular dystrophy that occurs in infants. Hemophilia A is X-linked recessive. A condition called retinitis pigmentosa, which leads to blindness, and usually children are able to see at birth, but then as they get throughout childhood and into adolescence, they go blind. And G6PD deficiency is a X-linked recessive condition, also quite common in PNG and related to malaria, as you know. And yeah. Okay. And then X-linked dominant conditions are much less common, and that in X-linked dominant conditions, males and females can be equally affected, because if a female has a 1X chromosome that's mutated and it's a dominant mutation, then they'll be affected. And the male, of course, is affected because they've only got 1X chromosome. So if they have the mutation, then they're always affected. They're relatively uncommon conditions, but hypophosphatemic rickets, which, well, it's not that uncommon in PNG. There's renal tubular acidosis and hypophosphatemic rickets can occur. Alport syndrome, which is a condition of genetic deafness and nephritis, if you see a child who's appears to have glomerulonephritis and has deafness, then they may have this condition called Alport syndrome. And again, it's an X-linked dominant condition. I talked briefly about mitochondrial DNA and how mutations later in life can lead to certain diseases like chronic cardiac disease or dementia, but there are also some genetic mutations in mitochondrial DNA that can lead to some conditions of infancy. Because they relate to energy production in cells, then usually children have quite severe and progressive developmental delay. They may have seizures. They often have a lactic acidosis because their Krebs cycle doesn't work, so their energy production comes often from anaerobic metabolism. They may have progressive neurological impairment, and when they get a minor infection, they tend to deteriorate. That's the phenotype of mitochondrial disorders. Again, you don't need to know too much about it, but just important to know that there is some genetic mutations that occur in mitochondrial DNA that can lead to this particular phenotype. That's usually the only type of abnormality. But if it occurs as a mutation in some organs, mitochondrial DNA that occurs later in life, then those organs might be prone to progressive impairment like cardiac failure or dementia, etc. So we talked about Mendelian inheritance, where there's single gene defects. I just want to point out that the majority of conditions that we see as being congenital problems in PNG are actually not single gene defects. They're mostly polygenic disorders, which are caused by an interplay of different genes and the environment, and sometimes teratogens, but not always. They're polygenetic, and we often don't know exactly what the underlying cause is, but conditions that like Hirschsprung disease or anorectal malformations, imperforate anus, relatively common in PNG, they're all polygenetic disorders. Occasionally, there's some rare Mendelian inheritance, like autosomal recessive Hirschsprung's disease, but most of it's polygenetic, and usually they're new mutations that have occurred, and the parents don't have those conditions. So there are some, as I say, teratogens involved with some of these anorectal malformations. There's some evidence of like some drugs, some sedative drugs, if a mother takes it in pregnancy, or these new antipsychotic medications called SSRIs that for sometimes for depression, or cigarette smoking in pregnancy can lead to some genetic mutations that can lead to anorectal malformations. They're very rare. Mostly the mutations that lead to these conditions are quite sporadic. They're not easily definable. There are forms of congenital hydrocephalus, forms of congenital hypothyroidism, and even autism are much more a condition being identified much more commonly now. There are 65 genes now known to be involved in autism, but also probably some environmental factors. I mean, there's some evidence now that, for example, like food coloring and food additives in highly processed food can lead to some changes, some mutations that lead to autism. So we'll know more about that later on. I wanted to briefly touch on gene regulation, and just to say that genes are regulated, like not all genes are turned on in a cell at any one time. And that's good, because that enables us to have specialized tissues, like specialized cells that become cardiac myocytes or become neurons or become alveolar endothelial cells or skin cells, whatever, because certain genes are turned on, therefore they make certain proteins and it transforms the cell into a certain cell line. But there are other ways in which genes can be up or down regulated, and that's called methylation. And when you add a methyl group to a nucleotide in a sequence, that inactivates the gene. And I don't want to talk too much about that, except to say that our genes are normally activated and inactivated, but also they can be abnormally activated and inactivated. And I want to briefly touch on these abnormal activation and inactivation of genes, and that's called epigenetics. And it means that certain environmental factors can turn on our genes or turn them off. I won't go through that in detail. I've talked about DNA methylation, but there's a number of other ways in which genes can be activated or inactivated. Many factors, some of which are hereditable, and some of which just occur in the individual. But many environmental factors, such as starvation or severe malnutrition or exposure to pollutants or toxins, can change our genes. And certainly there's good evidence of methylation in this gene called the AHRR gene, which is a form of epigenetic link to cancer, because that leads to lung cancer, squamous cell cancer, carcinoma of the lung. There are genetic origins of other forms of cancer. Even certain infections can lead to genetic changes that can increase or decrease our immune system. So mycobacterial infection, for example, causes these histone changes that lead to a turning on or off of the certain genes that may lower our immunity to mycobacterium bacilli, and that can increase our susceptibility to tuberculosis. So there's a complex interplay between environmental factors, between infections, our genes, and the turning on or off of those genes. There are certainly changes I talked about nutrition, and we know that from large studies of famines during in the Second World War in Scandinavia and Germany, there are changes that occur not only in those people but also in subsequent generations, and those changes relate to epigenetic changes that occurred in the genetics of people who had very limited access to food, and that can affect the mortality of even their grandchildren. So this is, I think, very important for understanding the early life origins of chronic diseases in adulthood. And we don't look at this enough, but certainly heart disease and type 2 diabetes and schizophrenia or other mental health disorders can relate to early origins, early changes that occur in epigenetics, in methylation of our genes, the turning on or off of those genes, and that can affect many things later in life. I'll just give one example of very adverse early life on events and their effects on epigenetic changes. And certainly many victims of the Holocaust showed DNA methylation changes, and their subsequent children and grandchildren did as well. We know that if a child's been prone to child abuse, if they've faced child abuse at a very young age, physical or emotional abuse, then they too will have changes in methylation in their genes that lead to changes in their stress responses, changes in their neural development, in poor physical health, and maybe even changes in their mental health later on that can lead to depression or post-traumatic stress disorder, or even other conditions. And that's linked to methylation of a particular gene called NRC31, which we know can be affected by early exposure to poor care or abuse, mental abuse or physical abuse, and it because of changes in the glucocorticoid receptor, which is a stress response gene. So there's very profound effects of early life events on people's genetic makeup and their genetic expression, their phenotypic expression later on, that can explain why some people end up with very difficult conditions later on. There's there's one more factor about genes that can affect expression, and that is what's called telomeres. And they're little sequences of DNA that occur at the end of chromosomes that protect our chromosomes from degradation. So they stop our chromosomes degrading as we get older. But these telomeres, they shorten with age. They're just DNA sequences, TTA, GGG. So thymine, adenine, guanine. They're just small DNA sequences, these telomeres, but they shorten as we get older. And it's been known that as telomeres shorten, they can be associated with certain conditions like obesity and diabetes and cardiovascular disease that of course, are metabolic diseases of adulthood. And certain exposure to certain conditions like stress or even loneliness or exposure to violence or in utero exposure to smoking or maternal depression or poor socioeconomic status, they can all have an effect on telomere length in the offspring. And that can lead to these metabolic conditions or these chronic conditions later on in life. So I think this is very interesting, this whole field of epigenetics, because it does help to explain why these conditions occur. Of course, they occur because of lifestyle choices as well, but there is sometimes a genetic basis that goes back to the very early life. Now, at the moment, the World Health Organization has a very strong push for more genetics being understood and used, I suppose, in low and middle income settings. And in Papua New Guinea, for example, the conditions in the global top 10 diseases that are relevant at a public health level, like thalassemia and G6PD. So you could argue what we should have is a diagnostic program for the early diagnosis of thalassemia and a program for the early diagnosis of G6PD deficiency, because it might help in the way we treat malaria, for example. There's a whole range of reasons why some early genetic diagnosis of these two conditions could be useful. I just wanted to finish now and just say why we should know about genetics. And even if we don't have sophisticated tests for many genetic disorders, then I think we still need to be good at genetic counselling, because that can be important for dispelling myths and stigma about certain conditions. It's not witchcraft or spirits or something. There is a basis for it that makes scientific sense. It can help us also understand the natural history of certain conditions and the prognosis of certain conditions. I think that's important, to understand the prognosis of certain conditions and to provide care for affected children and patients of all ages. There are adults who have genetic conditions, and as more and more children survive, then they become adolescents, and those adolescents become adults, and there needs to be a continuum of care for people who've got genetic disorders. I think it also helps us, understanding genetics also helps us to appreciate the multifactorial multifactorial disease causation. That is, we won't think it's all due to one factor, but understanding that it's often polygenetic is important. There's a whole range of microbial genomics that's happening at the moment, which we might not see, but actually is very important to understanding about resistance of bacteria and resistance of malaria or TB. And that's all based often on microbial genomics, which is a science that is becoming increasingly important for vaccine development and development of new antibiotics and things. I think we need to also appreciate the risk of eugenics emerging again. I haven't talked about this today, but in the last lecture I talked about the eugenics movement of the early 1900s. And I think if we have good literacy on genetics, if we understand genetics well, then we'll be much better as pediatricians at identifying when eugenics is emerging again. I don't think it's completely gone. And I think it's important that we recognize that. And there's very good uses of genetics, and there's also misuses of genetics. And if we have a good literacy of genetics, we'll be able to understand that. And lastly, so it helps us understand epigenetics. So the last types of conditions that I've talked about, especially the effects of environment in early life, environmental exposure on our genetic expression that might affect chronic diseases and things that happen to certain patients, certain people as they grow up and get older. So that's all I want to say today. That's a lot. And as I say, today's talk, I hope you'll be able to go back and look at last week's talk as well, because the two together are really, I hope, is almost all you need to know about genetics. All right, I'm going to stop sharing my screen now and stop the recording. And I'm happy to answer any questions if you have any.